So I wasn't sure if we were going to get to chemical digestion or not. So if we got through any of us, you can skip ahead to where we left off in lecture. But chemical digestion, this is when we are breaking the food down chemically. When food goes in, we have to break it down to its simplest forms so that we can absorb it. Again, that's the whole idea of eating, is we're getting food in, we're breaking it down, we're absorbing the nutrients we can from the foods we eat. So digestion itself is a catabolic process. Anything catabolic, catabolism means breaking things down. And so we're breaking down macro or large molecules into monomers, mono meaning one, like the single individual building blocks from those macromolecules. And they are small enough now that they can be absorbed. Things that are going to help in chemical digestion are enzymes. Whether these are intrinsically made, whether they are from various types of accessory glands like your pancreas, but we need lots of enzymes to break things down. Another thing that helps break things down chemically is water. It's a process known as hydrolysis. Lysis means break apart, hydro means water, is just we're using water to break apart bonds. And so again, it's also aiding in breaking down foods. So we're going to look at how each kind of food or each type of food is broken down, starting first with carbohydrates. Now, only monosaccharides can be absorbed, and when we say mono means single, these are single sugars. These are the smallest structures that are still considered to be a sugar. When we build things up, we put sugars together, we get disaccharides, polysaccharides, but only the single monosaccharides can be absorbed. Now, the basic ones that are absorbed, glucose, fructose, galactose, and so we need enzymes that can break apart your carbohydrates, because carbohydrates are made of these big, huge sugar molecules. Now, where we get some of these enzymes that break apart carbohydrates, the first place we see these enzymes is in the saliva. There's a salivary amylase that starts to break apart carbohydrates. There's a pancreatic amylase, so another enzyme made by the pancreas, and some of the brush border enzymes that are made in your small intestine. They're going to break down various disaccharides. They're going to try, if they can, to break down polysaccharides. A couple examples of disaccharides, sucrose, lactose, and maltose, is two sugars stuck together. Polysaccharides, your glycogen, your starches, they have to get broken down. My only note, cellulose, which comes from plant, is a carbohydrate. It's more commonly known as fiber, and although it is made up of sugar units, our digestive system, even with the enzymes that it has, can't break down those plant structures, which by fiber just goes right through you. Now, starch digestion does happen right away in the saliva, and the salivary amylase will break down various types of sugars and starches to what are known as oligosaccharides. These are smaller carbohydrates. They're not monosaccharides yet. And it works best, this enzyme works best at a slightly acidic pH. Pancreatic amylase that gets dumped into the small intestines breaks down any proteins, or not any proteins, any carbohydrates that have escaped salivary amylase, and breaks those also down into oligosaccharides. They're just smaller sugars. And brush border enzymes, you have what's known as dextrinase, glucoamylase, lactase, maltase, sucrase, are also going to, again, based on their names, break apart various types of sugars, and they will take a lot of these oligosaccharides, these already partially broken down carbohydrates, and break them down into their single sugars, those monosaccharides. And so you have carbohydrate digestion happening in the mouth, you have carbohydrate digestion happening in the small intestine from the pancreatic enzyme that dumps into the small intestine and that duodenum, and you have brush border enzymes that are coming from those brush border cells that line the small intestine, lots and lots of enzymes there that are breaking down some of these larger oligo or disaccharides down into their simplest monosaccharides. The next thing we have to break down, other than carbohydrates, is we have to break down our digest proteins. And so all of our proteins is all coming from our diet, but other places we find proteins, enzymes themselves are protein. Your mucosal cells, these are all eventually, these things will all help digest proteins down into their amino acid monomers, because proteins are made up of amino acids. And so we can break down proteins we eat, we can break down the enzymes we make, and we can even break down mucosal cells because they don't last forever and there's proteins in there that have to get broken down. One of the biggest enzymes that helps break down proteins is pepsin. It's one of those few enzymes that doesn't get ACE in its name. And pepsin works best at really low pH. That's where it's made, and like in the stomach. However, it's inactive once it gets to the duodenum. It likes that low pH of the stomach. And so it begins that initial protein digestion in the stomach. Now, we also have more enzymes further along that can break down proteins. We have pancreatic proteases. Proteases break down proteins, and we have trypsin, the chymotrypsin, and carboxypeptidase. Again, lots of things that end in ACE, but not all of them for enzymes. And they're eventually going to break down the proteins down to their single amino acid units. And then there are brush border enzymes that are coming from the cells that line the small intestine, aminopeptidases, which works on the amino side of an amino acid, carboxypeptidases, which work on a carboxyl side of amino acid, and dipeptidases that can work from either side. Either way, they're breaking apart even the amino acids. So we're breaking proteins down into amino acids, but we have enzymes that even break down the amino acids down into their single units. This is showing where a lot of this is broken down. Again, protein digestion does not happen or start until it hits the stomach. And from there you've got the pepsin, starts with the P for proteins, that it breaks down. And so it's going to start to break down those proteins into large polypeptides. Just means like a large chain of amino acids. Once that food gets to the small intestine, you have more pancreatic enzymes, those trypsin, chymotrypsin, carboxypeptidases. They're going to start to further break down those proteins, the polypeptides. And then really when it gets further down into the small intestine is we start to really break down these amino acids. And I might break them down into their individual amino acids and even start to break down the amino acids themselves. But protein digestion does not start until the stomach. Now, digestion of lipids. Well, I was going to say, generally, lipids are harder to break down. And so we do kind of a pre-treatment to our lipids to make breaking them down a little bit easier. We add bile salts. Now, bile salts themselves does not break bonds, but it does make these large lipid molecules into smaller lipid molecules. So instead of one big blob of lipid, now you've got small little blobs of lipids. And it's a little bit easier for the enzymes to then tackle. Now, top enzymes that break down lipids, anything that has a lipase in it. Lip for lipids, and ACE because it's an enzyme. And we've got pancreatic lipases. They help break down those lipids into fatty acids and into glycerol itself. It's one of the structures of a big fatty acid. And also it helps because we can absorb glycerol and fatty acids. We can absorb those structures into our blood supply in the villi, and then it can get transported to our hepatic portal vein over to our liver. Now, this is just showing that emulsification, we can take one big fat globule and using bile salts, break it down into smaller flat globules. We haven't technically changed the fat. It's still fat, it's just in smaller chunks. That's where the emulsification comes from. And it's these smaller chunks that the amino acids are going to work with to break it down, and then those various particles, some of the fatty acids, those can enter our digestive tract. We can absorb it. Now, where lipid breakdown happens? Well, it does actually happen, although I only talked a little bit about the lipases. And I'm like, you have lipases that start all the way up in the mouth. So we have a lingual lipase that right away starts the process of breaking down lipids. We have a gastric lipase that also is starting to help break down lipids. But it's really at the small intestine where you've got bile and those bile salts that are really helping do that emulsification, which does make breaking down lipids easier to break down, which then it makes it easier for absorption. So most fat is broken down in the small intestine just because of that emulsification process. But lipid breakdown does happen all the way back up in the mouth. Now, we also have to digest nucleic acids. It's it's the NA of DNA and RNA, and it's one of those, well, do you eat DNA? Well, if you eat things that were ever alive, and you do, they have DNA in it, so we have to break that down. Plants have DNA, any animal meat you eat has DNA, so it has DNA, so we have to be able to break down the nucleic acid of what you eat as well. So we have more enzymes, mostly coming from the pancreas as well as parts of the small intestine. We have a pancreatic ribonuclease that breaks down RNA, and we have a pancreatic deoxyribonuclease that breaks down DNA, and it breaks it down into its nucleotide monomers. So we're going to be breaking it down into your bases, your sugars, into your phosphates, other things that help do that as well. Other than the pancreatic enzymes, lots of enzymes come from the pancreas. It's why you cannot live without the pancreas. Is the cells that are in your small intestine. You have got those brush border enzymes called nucleosidases and phosphatases that further are going to break apart your nucleotides. So lots and lots of breakdown of DNA and RNA. So it doesn't start till it hits the small intestine, because it's that pancreatic enzymes that dump into the duodenum and the brush border enzymes that are found in the small intestine that break those nucleic acids down. Now, we've been talking a lot about breaking things down, but that's only the first part of digestion. We have to break things down, then we have to absorb them. And so almost all of your food, about 80% of your electrolytes, and most water is absorbed in the small intestine. So, most of it's even prior to the ileum. So you've got the duodenum, jejunum, the ileum. So most of all of this is all absorbed before it even gets to the ileum. The ileum is really there to try to reclaim some of those bile salts to be able to reuse them. So they say, only maybe 1 liter of 10 liters of food, maybe about 10% of what you actually eat makes it to the large intestine. Everything else is broken down and absorbed. Most of it's absorbed by active transport, meaning it requires ATP and into the blood supply. A couple exceptions, lipids are passively moved through. And we just create a nice gradient with lots and lots of fatty acids next to areas that don't have a lot of fatty acids, and they passively move through. So we talked about breakdown of carbohydrates, lots of enzymes, and we want to break the carbohydrates down into those monosaccharides, those simple sugars. Well, now we want to absorb them. Glucose and galactose are absorbed through what's known as secondary active transport, meaning or co-transport. So they really are moving them. We're using ATP, it is an active transport, and we are moving these sugars, and using ATP we are transporting them with sodium as well. So we are going to actively pump glucose and galactose out of the small intestine into epithelial cells with sodium. Now, they can also move out of epithelial cells by facilitated diffusion and eventually make it into our blood supply. So we're going to pump them out of the digestive tract, and then they will eventually, through facilitated diffusion, make it into our blood supply. Fructose is going to use facilitated diffusion to enter and exit cells. So it doesn't get transported or co-transported with sodium. But the sodium becomes important when we're moving sodium around. We can also absorb proteins. So we broke proteins down, we digested them down into their simple amino acids, and they're also transported by different types of carriers. Most are also coupled to active transport of sodium. So as we are pumping sodium somewhere, we can also be pumping amino acids, and vice versa. We can actively pumping amino acids, and then we'll let sodium kind of tag along and go with it. Now your dipeptides, which means 2 amino acids, or tripeptides, which means 3 amino acids, are actively transported by a hydrogen ion dependent cotransport. Meaning when we're pumping hydrogen ions, things where we need something to be acidic, we can be pumping some of these larger protein molecules. Let's say they're large proteins, large amino acids, because there's 2 or 3 amino acids stuck together. And then they'll eventually get broken down into their single amino acids in the epithelial cells. So we're going to pump these, 2, 3 unit amino acid molecules into the epithelial cells where they're going to get further broken down. And then they enter the capillary blood by diffusion. They want to go from an area where they've got lots of amino acids to an area where there's little amino acids. We also want to absorb those lipids, and so same image as I had on there before is that we are going to break apart the lipids. We're going to break it down into your fatty acids and your monoglycerides, and I'm like, it's glycerol. And it forms kind of these little tiny structures called micelles as we break it down into these units. Now, these micelles can get pumped into your epithelial cells, and they're going to enter these lacteal structures in the epithelial cells, and then they get transported to your cardiovascular system. And it's there that we can use those lipids for energy. And I'm like, and if you don't need it for energy, we store it as fat. And then the last thing we talked about breaking down was nucleic acids. So we broke them down into our simple structures, and I'm like your simple monomers, your bases, your phosphate groups, your sugars. Well, we can also absorb them. It's an active transport. It's a pretty easy active transport, which is why I don't have more information on here. It's just we're using ATP to pump these, as I say, monomers of nucleic acids across the epithelium into the bloodstream. Now, one thing we didn't talk about breakdown, and I'm like, was vitamins, but yet we still absorb vitamins. We just don't break them down. They're ingested or made as already their simplest unit. So we're not breaking them down, but we still absorb them. In the small intestine, fat-soluble vitamins such as A, D, E, and K are carried with fat. And I'm like, they're carried by those micelles and then diffuse into various cells that are needed. And so if you take various vitamins that have A, D, E, and K without eating foods with fat in them, little or no absorption of those particular vitamins actually takes place. And I'm like, some vitamins have to get absorbed with those fat molecules. We also have water-soluble vitamins such as C and B that are absorbed by diffusion, or they could be absorbed by passive or active transporters. So lots of different ways they can get absorbed. Vitamin B12, we talked about this, it comes of the stomach, binds with that intrinsic factor and absorbed by endocytosis. So if you don't have the intrinsic factor, vitamin B12 will never be absorbed. Now, most absorption of vitamins takes place in the small intestine. A few absorption of vitamins takes place in the large intestine, and that's because the bacteria that are in the large intestine can make some vitamins, and if they're made in the large intestine, they're going to be absorbed there. They can't move their way backwards to the small intestine to get absorbed. So vitamin K and various vitamin B vitamins that are made in the large intestine can get absorbed in the large intestine as well. We can also absorb a lot of the electrolytes, a lot of these charged ions. So most ions are actively transported or actually absorbed along the length of the small intestines. So a lot of things in the small intestine. Iron and calcium are absorbed in the duodenum and only in the duodenum. Sodium is usually coupled when it's moving glucose and amino acids, as we already mentioned. Chloride is usually transported actively, usually when you're moving bicarbonate ions. Potassium diffuses in response to osmotic gradients. And I'm like, which means, and I'm like, if you've got rising levels of potassium somewhere, it's gonna want to go where there's less potassium. And so I'm like, it just moves where there's less potassium. It usually is also affected by your water absorption. So if water is absorbed from the inside of the intestine, the lumen of the intestine, you're going to have lots of potassium left over, and that will cause potassium to move. And I'm like, if that changes in absorption, not as much water is absorbed, leading to not a lot of potassium. It's not going to want to move. So it just diffuses when water moves it. Usually the amount that's absorbed in the intestine, I read that wrong when I was just reading it. I'm like, usually whatever electrolytes are in the intestine is how much is absorbed there. We want to absorb what's there. We don't want to absorb, well, you can't absorb any more than what's there, but pretty much we want to absorb everything back that we possibly can. Our body tries to recycle and reuse as much stuff as possible. Now, some things that can change with the absorption of electrolytes: iron and calcium. Even though what's usually there is what's usually absorbed, iron and calcium can be absorbed related to the need. And so if you need more iron can be absorbed. Now, iron ions are transported in the blood by a molecule known as ferritin. It's a protein. And calcium absorption is regulated by vitamin D in your parathyroid hormone. That's the hormone that releases calcium from the bones if you don't have enough calcium in your diet. And so how much calcium is absorbed is regulated by those. And again, calcium is regulated by vitamin D, which is why most milks, and I'm like, are vitamin D supplemented? Now, we also want to absorb a lot of our water. About 9 liters of water, mostly from all those secretions that your digestive tract made, get absorbed every single day. So that's about 95% is absorbed, and it all happens by osmosis. Water wants to go where there's not as much water. The rest is absorbed in the large intestine. Not all of it is absorbed in the large intestine, and like some of it is left in that fecal matter just to soften the feces. The net osmosis occurs as long as you have a concentration gradient established. You have lots of active transport. When we talked about lots of things being absorbed, active transport. It required ATP. We used energy. We pumped various types of molecules across the digestive tract. Well, if you pump lots of molecules across the digestive tract, water wants to follow. That's the thing. Water always wants to follow the solute. And so water uptake is always coupled with solute uptake. So water follows solutes. We used ATP to pump solutes, water's going to follow. Now, there's some things that can happen though. So, we always have problems, and it's malabsorption of nutrients. If for some reason you can't absorb some particular nutrient, it can result from anything that interferes with your bile itself, pancreatic juices, anything that interferes with breakdown of food and absorption of food. And a big one that a lot of people have heard of, it's known as gluten-sensitive enteropathy, also known as the celiac disease, which can affect about 1 in every 100 people. And it's because when they try to absorb the protein gluten, which is found in all grains except corn and rice, and I'm like, when they try to absorb, that's an absorption to that protein, there's an immune reaction. That our immune system recognizes that particular protein as a foreign body, and we have immune system that's going to try to fight it off. And so that particular protein is going to cause immune cell damage to the intestinal wall, to the brush border cells, and really there's no way to fix it. The only treatment is just don't eat gluten in your diet. So you have to eliminate all grains except rice and corn. So I'm like, oh, no wheat. So I was going to say, I'm like, it's just having to do with absorbing that particular protein. Now, since we're done talking about digestion and absorption, just some developmental aspects with your digestive tract. So fetal nutrition before the baby is born all happens from the placenta. However, the digestive tract is working even inside the female. It's stimulated to mature because amniotic fluid is swallowed while inside the uterus, and that fluid still is, training the digestive tract to recognize it, break it down, and create additional fluids. Other developmental aspects, the newborns have a rooting reflex that helps them find a nipple, and a sucking reflex that aids in swallowing. Again, and I'm like, they're born knowing how to eat. And I'm like, because it's the most important thing that they need to do. Now, newborns double their birth weight in 6 months. And I'm like, which, yes, if you've ever seen a baby and then seen it 6 months later, I'm like, yep, they double in weight in 6 months because of all the nutrients they're getting from mom's milk. And by the time they're an adult, and I'm not an adult, by the time they're 2 years old, they're eating an adult diet. Pretty much means they can eat anything an adult can eat. As we start to get older, and we're jumping from 2 years to middle age, and I'm like, we can start to develop some digestive issues. Usually it's ulcers, cholecystitis, this is gallbladder issues, and I'm like, it just start to have more and more issues. As we get even older than middle age, your digestive tract activity starts to slow down. There's usually less digestive juices. Absorption of food is less efficient. Peristalsis slows down, and you have less frequent bowel movements. Now, the problem that comes from that is if you have less frequent bowel movement and that peristalsis slows, feces hangs out in the large intestine too long. And that's when you can get the constipation, is because too much water is absorbed. It's why you see things like prune juice and fiber supplements always is targeted towards the elderly, because their digestive tract is slowed down, which leads to constipation. Also, your taste and smell becomes less acute. Sometimes you just can't taste anything or smell anything anymore. Periodontal diseases in your mouth become more prevalent. You can suffer from diverticulosis. We start to get those diverticula or kind of bulges on the intestine. You can have fecal incontinence, might not be able to control that external sphincter anymore. And any type of cancer is going to increase of the digestive tract. And so stomach cancer, colon cancers, all of those usually start to get older. They're going to start to recommend the colonoscopies to be checking for any abnormalities. Now, some review in terms of gastrointestinal function. The large intestine's greatest contribution is to absorb water. It's not absorbing other nutrients that happened in the small intestine. It doesn't have anything to do with chyme or pancreatic enzymes, all the small intestine. And gastric motility, also small intestine. Which of the following enzymes breaks down lipids? And it's none of them. The enzyme that breaks down lipids is lipases. And so I'm like, there's the end. So again, if we get to any part of this, hopefully you skipped ahead to wherever we didn't get.